first, let me just say it's a pleasure to welcome you to the 2024 Charles L. Cooney Lecture at MIT. This lecture was established just 2 years ago in 2022 as a premier showcase for rising stars and young talent in the biotechnology arena, bringing emerging leaders in both the academy and industry to campus to discuss aspects of innovation in biotechnology. And of course, this year we're featuring someone who's coming to us from the industrial sector. Before I introduce the individual for whom the lecture is named and our honored lecturer for the afternoon, I'd like to say just a few words about our sponsor, and that would be our very own alumnus, Noubar Afeyan. Noubar entered biotechnology during its emergence as an academic field and as an industry, completing his doctoral work in biochemical engineering here at MIT in 1987. During his career as an inventor, an entrepreneur, and a CEO, Noubar has co-founded and helped build over 50 life science and technology startups. These companies include, of course, Moderna, Kodiak, Denali, and many others. He's written numerous scientific publications, and he's an inventor on more than 80 patents. He's a lecturer at Harvard Business School, and from 2000 to 2016, he was a senior lecturer at MIT's Sloan School of Management. He teaches and speaks around the world on topics ranging from entrepreneurship, innovation, economic development to biological engineering, new medicines, and renewable energy. He's a member of the MIT Corporation, and as many of you will remember, he was our commencement speaker this year and delivered a really thoughtful and I would say quite timely speech that everyone there really appreciated. We were quite proud to have ChemE represented, and I did talk a lot of smack on stage about that, Noubar. And now to introduce the person for whom the lecture is named, Charles Cooney. Charlie is Robert T. Haslam Professor Emeritus here in our Department of Chemical Engineering. Charlie obtained his bachelor's degree in chemical engineering from the University of Pennsylvania, his master's degree and PhD in biochemical engineering here at MIT. After working briefly at the Squibb Institute for Medical Research, he joined the faculty of MIT as an assistant professor in 1970, and he's been a full professor in the department since 1982. He's received a lot of awards, more than I could possibly name. A few are the 1989 Gold Medal of the Institute of Biotechnological Studies, the Food, Pharmaceutical, and Bioengineering Award from AICHE, and the James Van Lennon Distinguished Service Award from the American Chemical Society's Division of Microbial and Biochemical Technology, which we call BIOT. And he's a founding fellow of the American Institute of Medical and Biochemical Engineers, as well as a fellow of the American Chemical Society. He's been a consultant to multiple biotech and pharmaceutical companies, and Charlie sits on the boards of Genzyme, Polypore International, and Biocon in India. His research and teaching span many aspects of biochemical engineering and pharmaceutical manufacturing. He holds more than 30 patents. He's published more than 250 research papers, and he's co-authored or edited 5 books, including Development of Sustainable Bioprocesses, Modeling and Assessment, published just in 2006. In addition to his professional interests, Charlie is a trustee of the Boston Ballet, an overseer of the Boston Symphony Orchestra, and a board member of MIT's Community Service Fund. Other interests include rock climbing, skiing, scuba diving, antique map collecting, and a very picturesque hobby, which is high-altitude mountaineering. And he has ascended Denali, Amand Dablam, Mont Blanc, Kilimanjaro, and Huascarán. And these are pictures that Charlie provided to us that show him approaching the summit of Amand Dablam in Nepal at 7,000 meters, and then Charlie with his son Matt on a summit with Mount Everest in the background about 10 miles away. We know that Charlie has always been at the forefront of biomanufacturing and has really been involved in thinking about how we advance beyond our existing paradigms into new ones, including in continuous biomanufacturing. And so this is a new slide from last year because this was just announced that next month Charlie will actually receive the inaugural Lifetime Achievement Award sponsored by Sanofi from the Integrated Continuous Biomanufacturing Conference. And I'm going to go off script for a little bit and just tell a little vignette. My mother always told me you should tell people the impact that they had on your lives while they're around to hear it. And Charlie probably has forgotten this, but 30-some years ago, spring of my junior year, Charlie was teaching biochemical engineering. For those of you who are much younger, there was a world before there was the internet. And I went to talk to Charlie and said, I'm interested in going to graduate school, and I want to know I'm interested in biochemical engineering, and I want to know what are the places and schools that I should be looking at. He took way more time with me than I thought he would. And then he said two things to me that actually changed my life. One is he said, if you'd like to do a UROP, you're welcome to do that in my lab. And if you would like me to write you a letter of recommendation, I'm happy to do that as well. For a 19-year-old kid from East Texas who didn't really know what it meant to say she wanted to go to graduate school, it really did have a profound effect on me and is a big part of why I am able to introduce the Charles L. Cooney Lecture today. So now let me introduce our speaker. Jordi Mata-Fink is co-founder and chief executive officer at Gate Bioscience. Gate is developing a new class of medicines called molecular gates, which are small molecules that selectively eliminate harmful extracellular proteins at their source, which is inside the cell. Since its founding in 2021, Gate has raised nearly $70 million from leading life science venture capital firms. Gate is based in the San Francisco Bay Area and employs 30 scientists, engineers, and drug discoverers. Jordi began his career at Flagship Pioneering, a premier life science venture creation firm, beginning as a summer fellow while still a PhD student. At Flagship, Jordi helped originate and launch multiple biotechnology companies, including Editas Medicine, Rubius Therapeutics, Signal Therapeutics, and Generate Biomedicines. Jordi earned a PhD in chemical engineering from MIT in 2012, where he worked with Dane Wittrup to apply the tools of protein engineering and directed evolution to the design of HIV vaccines. He was an inaugural fellow of the Reagan Institute of MIT, MGH, and Harvard, and he was an NSF graduate research fellow. Jordi received a BS and MS in chemical engineering from Stanford University, and he now lives in Portland, Oregon with his wife and 3 elementary-age boys. And when he's not leading Gate, Jordi enjoys hiking, skiing, and coaching youth soccer. Jordi and I had a phenomenal conversation this morning, and I know you are really going to enjoy this lecture. It is a pleasure to have you back, and I'm happy to turn it over to Jordi Mata-Fink.
We all set? Thank you, Chris. How's the audio? Good? Thank you for that introduction, Chris. It was really very wonderful. And to the faculty, thank you for inviting me to come back. After some of the GSC-X skits in my time, I didn't know if this would happen. Charlie, it's a real honor to be here giving this lecture in your name, for everything you've done, and Nubar, for everything you do to promote the next generations that come after you. Thank you. All right, I thought I'd give this talk for the students and postdocs in the audience, and thought I might tell you a little bit about my journey to where I am today, and some of the lessons along the way, things that have gone well, things that, mistakes, with the hope that perhaps some of those will help you as you embark on your journeys. I don't quite have the archetype of the technical entrepreneur journey. It's not that my research project turned into a company that I then led. But perhaps that's more common. The non-archetypal journey might be more common. So let me tell you where I've been, and then we'll sort of jump into some of the stories. As Chris was saying, I started at Stanford. I came here. I worked with Dane on vaccine design using the tools of protein engineering. Ended up walking down the street when I finished and worked at Flagship Ventures at the time. It was actually a really fascinating time at the firm. Nubar was shifting it strategically from an early stage venture capital company that did some company building to a venture creation firm that was all in on building their own biotech companies from the ground up. So I was very fortunate to get there at that time and be part of the first wave of Flagship figuring out how to do that process. I spent 8 wonderful years at Flagship. Over the course of that time, helped to start 4 companies. So Editas Medicines was the first of the CRISPR-Cas genome editing companies. Rubius Therapeutics we did with Harvey Lodish and Hida Pluch, who were at the Whitehead Institute at the time. This was red blood cells as a cell therapy. Signal Therapeutics was looking at peripheral nerves and their interactions with the immune system. And then Generate Biomedicines, which was in the first wave of applying the power of machine learning and artificial intelligence to protein structural biology and the generation of new protein therapeutics. So for family reasons, we moved out to the West Coast. COVID came, the disruption we all remember. And after a bit of time off, decided to roll up my sleeves and build a company called Gate Bioscience, where I'm currently CEO. And as Chris was saying, we'll talk a bit more about this along the way. So GATE is developing a class of small molecules that eliminate disease-causing proteins at their source inside the cell. So the first lesson I was thinking about in reflecting on this is actually one that predates my MIT time. And I learned it from this guy, Steve Jobs. It was actually my last day on campus as an undergraduate at Stanford. Steve Jobs was my commencement speaker. So this was at the time he had gotten kicked out of Apple. He'd then gone and done Palm and Pixar, got brought back into Apple when the company was falling apart, rescued them. The click wheel iPod had just been released, the iMac, the first generation of the MacBook. And so he was really on the rise, but also had been quite private about his life. So he hadn't really told his story, and this was his first sort of public telling of his story. And he told something really interesting. When he was a college dropout, he was telling us this story, he was up at Reed College in Oregon, bumming around on some friends' couches. He audited a calligraphy class, of all things. And he talked about how in that moment he learned how letters need to fit together asymmetrically in order to be pleasing to humans. And that he used that lesson later when he and Woz started Apple to actually build a human-computer interface that was pleasing. And that was what made Apple a technology company that made devices that people loved and not just devices that people used. Really profound, so this connection. And then he stopped and he said, you can only connect the dots looking backwards. That there's nothing in that moment as a college dropout taking calligraphy classes that would have predicted the ability or that future path. And so that concept really stuck with me. I think of it as sort of finding the best fit line of your life, right? As you think about the choices ahead of you, it's always possible to look back and, depending on your skill as a storyteller, to tell a really good story about how everything led to where you are and why it was important for each of those steps. But when you're looking towards the future, you really can't draw that best fit line. So the lesson for the students and the postdocs that I would share is that if you're sitting in these chairs as I was about 18 years ago, having no real idea what I wanted to do with my career or really how to get there, that's perfectly normal. And that's part of life, and you should embrace that feeling of uncertainty. So lesson number one, if you're drawing that best fit line, you can only do that looking backwards. But you are going to have a lot of choices because you're very skilled and you have lots of things you could do and opportunities. So how should you think about picking what to do? This next lesson, also somewhat general, I think of it as a lesson in fugacity. And I'll get back to that. Because there's an infinite number of things you could do. Right? The number of roles available, not just in biotech, but in any career, is far vaster than what you can imagine sitting here today, not counting the roles you might create for yourselves. But what I've seen over and over again, experienced then with the people I've hired and worked with, is that the most important predictor of whether or not you're going to succeed and thrive and be happy is if your internal drivers, whatever they may be, align with what the role needs and asks. If you can fit those things together, you'll find yourself happy and motivated and in a healthy spot. And this is under your control. It's finding your thing. I think of it as fugacity because it's sort of it's not the skills that you have. I'm not suggesting only do things that you're already good at. That's not the advice. It's that, do you ever have that moment or that experience where you sit down to do something and then you look up a minute later, but hours have passed? Right? That state of flow that you can get into. Things that you do that when you're done doing them, even after a full day, you have more energy than when you started. That's what I'm talking about. That thing. I think the fugacity, the kick to get you free from whatever state that you're in into that next state. It's worth finding that out about yourself because that's what you can use to then figure out whether those things that are true about you align with what roles may offer. And to be clear, those will be different for everybody. And even within a single role or title, they can be different. So this is a photo of my two co-founders at GATE. So we all have the same title of founder. But I can tell you that Pat, the gentleman on your left, his thing is molecules and puzzles. He can sit down and think about how a chemical structure is going to bind and move it around, and he can spend hours and days and he thrives on that. That's his thing. And Roman in the middle, so he is a people and what they want and how they fit and how he can put them together and sort of, motivations. And that's his thing. He could sit and think about people and how they fit in organizational charts for hours and days. And for me, it's storytelling and it's energy of small teams. and finding ways to put those two together and sort of anything that involves gathering people together and pointing them in a direction or steering in a direction is just, that's the part that I can spend infinite time on. There's a book that's actually helped me along the way, this book called Designing Your Life by Bill Burnett and Dave Evans. They're professors of design at Stanford, and basically what they do is they take principles of design thinking to the question of what you should do with your life, a question that many undergraduates struggle with. It's quite useful. Some of the early chapters actually have exercises for how to go through and figure out what are the things that give you a state of flow and that give you that sense of freedom. But perhaps I recommend you get the book and work through it, but there's maybe a bit of a shortcut, which is to think about where you spend your time when you have unfettered time, because that often is pointing you to things that bring you joy and give you that sense of freedom. And so to go back to fugacity, for me, when I think back to some of my time at MIT, I distinctly remember early in 1040 having a problem set with fugacity of species C in the liquid phase and being stuck on it and doodling around it and coming up with that and saying, gee, that looks like a soccer team logo, like FC Barcelona, and hey, FC Landau, and we started the FC Landau first year chemical engineering intramural team, which actually is my only award at MIT, was the Rock Award, and it was for that, incidentally. But looking back in hindsight, it's clear what I liked, what gave me the joy and the freedom and the escape was small groups of people and motivating and bringing together. So I encourage you to figure out what those are for you and that they may be hiding in plain sight. Right? All right, that's enough kind of general life lesson. Let's get back to the path. So I was working with Dane, getting close to the end of my PhD. Let me tell you about how I got to flagship. It's going to be a lesson about Arrhenius equation and reaction rates. So that's Svante Arrhenius on your left, and on your right is this fellow named Erdene Beshemov. So Erdene was an MBA student at Sloan, and I'd gotten to know him. He had started a publication out of Sloan called the MIT Entrepreneurship Review. Now at this time, there wasn't really the infrastructure quite as established as there is now. You know, the biotech club and the various resources available for connecting entrepreneurs at MIT. And Erdene was trying to build this. And he was going out trying to find grad students with some spare time to write articles about entrepreneurial activities taking place at MIT. So a bit off the beaten path, but I got to know him. I said, sure, I'll write an article. Tried it out, and we got to know each other. And then he ended up at Flagship. And one day he called me up. He said, Jordi, I'm at Flagship, and they have this internship, and it's going to be great. I don't really know what it is, but you're going to love it, and you should sign up to do it this summer. So let me tell you a bit about my PhD journey. It was a bit of a struggle to get to a project that had progress and sort of, you know, some of you may remember that feeling or be living that feeling. And this was the moment in time when I finally had line of sight to the end of my PhD. I had Erdene saying, you got to do this internship. Yeah. I'm thinking, no, Erdene, I've been 6.5 years and I finally see the finish line. I can't. But he convinced me. I went and talked to Dane. You remember this? I said, I think I want to do something in startup world, but I don't really know what that is. And I don't know how, this Flagship thing seems like it could be a way to figure that out. And Dane said, well, you may not graduate quite on the timeline that we've talked about, but if this is what you want to do, by all means, go do it. So I went to Flagship, and so the rest from there we'll talk more about in the next story. But you know, in thinking about reaction kinetics and the Arrhenius equation, there's 3 parameters here that you can fiddle with, right? One is temperature, and we're at Kendall Square in 2024. The temperature's very high already, right? So we're fine. You can increase the frequency of your collisions. Put yourself in places to meet people like Erdene and bump into them and other interesting characters who can open doors. And you can lower the activation energy, which means when Erdene says something like, hey, go write an article for me, or hey, you should try this internship, you say yes. Those are the 2 parameters under your control. And so I would encourage you to, as much as you can, increase the frequency of those collisions and decrease the activation energy. So say yes, and especially when saying yes makes you feel uncomfortable, those are usually the ones that open the best opportunities. Good, checking in on time, perfect. So I got to Flagship. I moved from the academic setting into startup land. And if you guys don't mind, I'm actually going to switch to a chalk talk for just a little bit, because what I discovered when I went into industry is that it felt somewhat like a phase transition, in that the rules were different. It was like a different place, there were different dynamics. And fortunately, as chemical engineers, we're very good at taking a complex system and simplifying it down to its governing parameters and figuring out what those are. So I thought maybe I'd just sort of map for you guys thank you. We got value on the y-axis, we got time on the x-axis. That's basically the curve of any project you're working on in a company, of value versus time. So okay, what is value in this setting? If we're talking about a mature company, you might say, okay, this point here, value is some new idea, proof of concept. And maybe here it starts to look like a drug. And here it works in people. And here is a standard drug development type of value inflections. But the same dynamic is true sort of like in a fractal sense at any scale you look at. So in the case of a very early stage startup, this may be some initial result that gives you some sort of aha moment. Value on the y-axis can be one of two things. It's basically either de-risking of the idea to make it more likely that it's going to work, or increasing the size of the opportunity at the end of the day if it does work, right? So if you're an employee at an early startup, you need to be thinking about what are the experiments I can do that increase the value of the thing I'm working on. This is different from the academic setting where the y-axis tends to be novelty, right? Bringing money in the academic lab is about getting grants funded and publishing papers. And so that novelty is what drives that. But in this case, understanding that it's value, not novelty, what value is, and importantly, how that relates to time. That's the dynamic that I experienced in looking at sort of the startup world. I remember at one point in my PhD, I could have ordered a gene block oligo for probably $1,000, and instead I said, I'm going to buy the PCR primers and I'll just assemble it myself. Because my time didn't have any value. But I tell that story to every new employee who comes into a company, especially the ones coming straight out of grad school. That 3 weeks or 4 weeks that might take you to do the PCR, if we're burning $200K a month or $1 million a month or $2 million a month, no, spend $1,000 on the gene block, right? So understanding time and value, understanding where those inflections are, understanding actually that the inflections, you may have to convince someone of that inflection is valuable. Right? If it works in people, sure, everyone says they understand that. But in the earliest days, you have to be able to convince someone why the advance or the insight you made is valuable. So I think this is a dynamic that's very important to understand. The other piece is money, right? So you need money to fund this. So you could raise money and just do all the work. You can't actually, it's too expensive, right? If you're going to take a drug all the way through approval, you cannot raise that money in one go. It's too much money, it doesn't exist. But there's another reason, which is the money's too expensive at that point. So if this makes sense as a concept, but when you're raising money in a company, you're actually selling shares in the company for that money, right? The transaction that you're doing is you're selling shares in the company. And the number of shares you sell for that money depends on the value. So if you're very early on in your journey and your company's worth $1 million, let's say, and you need to raise $10 million to do your experiments, well, now the company has $11 million total, but $10 million of that's owned by the investors. You, as the founder, own a little bit under 10%. Right? Whereas if your company's worth $10 million and you raise 10, now you own 50% after that transaction. Right? So the dilution, if you were to try and raise all that money right here at the beginning, not only would no one give it to you, but also you would dilute yourself entirely. So you might say, okay, that's not the way to do it. Let's be more efficient. Let's do like a trapezoidal rule. Just raise the money as you go. Every time there's an incremental value increase, raise some more money. Can't do that either because it takes time. You can't sort of every single day go out and add a few extra pennies to the coffer. But what you do have to do is you raise money in steps. Right? So each time you've made some value increase, you then go out and raise money for the next phase. And now you're raising money where your value has increased, and so you're not giving up as much of your company for the next round. But this starts to matter because when you go out to raise this money, what experiments do? You need to find ways to bring this value curve to the left, right? Get as much done to get the biggest value increase you can before the money runs out. Investors on the other side of the table are thinking about the same curve. And they're thinking, oh, you're asking me for this amount of money. Are you going to get to something valuable? What if I gave you more money? Would you get more? And if I give you this money and you run out and you're still down here, the next round's going to come in and I'm going to get diluted. Right? So this is that dynamic of increasing of value and how it relates to time that basically governs much of how venture capital works and biotech fundraising works. I encourage you to figure out the rules of whatever system you're in. The rules will change. Be curious about them, and you'll be able to figure them out, and that's going to be really powerful for you. Thank you. So how does Flagship fit into this model? Actually, interestingly, one of the things that Nubar figured out, and as I told you, when I joined Flagship, it was at this transition moment between a more conventional venture capital firm of the type that we just mapped out and a venture creation firm, where essentially, you can keep the screen down, it's all good, Flagship invents the technology, but also puts in the money to support it. And so now what that lets you do is a couple of things. One is you can build more value before you have to go out and ask other people to put money in, and so you can have more of an ownership. And I got to say, the Flagship model has become quite popular and is often copied, I think because people look at the economics and say, that's a better way to make more money on this. It is nowhere near as simple. Investing money earlier is not the secret. The actual act of entrepreneuring, of building that value in the company, I think is really, that's a different skill than just investing early. But the other thing that this type of model, the Flagship model, where you own the company all the way through and sort of put the money in, is that it lets you take bigger creative leaps in developing the companies. When you're building a company in this model, it's really hard to think beyond the next round of fundraising. You're always thinking, what do I need to do that's going to be fundable by the next set of people putting in money? And if this is something like 2 to 3 years away, which is the typical life cycle for funding rounds in biotech, you're always thinking about what can I do in the next 2 to 3 years that will be convincing to other people who are in the market to put in the next set of money. And that's very constraining on creativity. It forces you into adjacencies to what's known already. And so I think, at least in my experience, and we have Nubar here, and he can correct me with different opinions, but that one of the magical things about Flagship is the freedom that it gives the entrepreneurs within the firm to really think about more ambitious technological leaps, and to be able to do that with the freedom of not having to go raise money 2 years from a market that may shift with time. Let me tell you just briefly about some of what that special sauce is of how to get that type of creativity and to do so in an institutional setting. So one of the things, maybe I'll just share just of one concept here, which is exploring the landscape first. So it's an optimization problem. If you start with an idea, there could be sort of an infinite number of ways you could go with that idea in the future. One of the things that Flagship does particularly well is to map the future world and figure out if some place is worth going to before you build the company to go do it. If you have the ability to sit before you've committed resources to a project, you have the intellectual freedom to think about where do you want to actually end up. So I think of it like mapping a potential energy landscape or other surface to try and figure out where you should go. Okay, a couple of things. Ideas, super abundant. Coming out of my PhD, I remember thinking, well, the thing that I've been working on for so long, it's my baby, I can't give it up. Often the entrepreneur who comes with a technical idea thinks that that's the only good idea they'll ever have, or it's the one the sunk cost of putting all the effort into developing that idea. Ideas are abundant. So worth setting aside initial concepts coming in. For example, you say, here's my tool, let's use it for this problem. That may be the best problem for your tool, but if you shift your frame of reference and you're sitting at the problem, is that the right tool for the job? When you could pick from any number of tools. Right? Run thought experiments. They're much cheaper than real experiments, and faster, and you can be creative with that. And try to do so untethered from what's true today, or what is constraints of what's available today. Because if you only imagine things that are doable with things that exist today, you're only going to get within the tether of what's available today. So the thought experiments, you just have to sever what you know today and imagine that future, you might find a destination that says, that's a really valuable place to go. Now let's go figure out how to build the steps to get there. Right? But you'll only be able to do that if you first imagine the solution. You can sit in an office by yourself and do thought experiments, or you can go talk to people. And talking to people is an incredible multiplier, a force multiplier of creativity. Talking openly. When I first started, I used to think that, well, the idea, the moment I tell it to somebody, they'll compete, or I'll lose, or so there'll be some sort of competitive disadvantage. No, it's all in execution. It's all in execution. People love to talk about ideas, be part of the community, talking openly about what you're thinking about. That's the way to get that sort of reciprocal openness and to learn things, and then you can decide where you're going to point your engine and chase. And then the final bullet point, so what, is so much more important than what. We're very good as engineers of describing what it is that we're doing, but the height of that potential energy landscape, or the depth, I guess if we're doing the inverse, is so what. It's the value proposition of those ideas. A little shorthand that I learned from Avak Kavedjian, who was one of my mentors at Flagship. Your technology will have features, what it can do. Those features will give rise to advantages. They're better than other ways of doing it, which will ultimately turn into benefits to somebody. The value proposition, what you have to describe, is the benefit tied to that advantage that comes from the feature. It's the triad of those together. It's not my thing can do this, it can do that. This is better than that. It's better. No, it's the combination, the benefit that comes from the feature and the advantage. So you do that process of exploration. That's sort of, if there's one lesson I can share, Nubar, from my time at Flagship for everybody, it's this, of exploring the landscape first. And you can do that with your ideas. There's nothing stopping you from running these types of thought experiments and talking to people, even where you sit today. So I get to Flagship, and I think it was about 3 weeks later that the first CRISPR papers hit. And it was an incredible stroke of luck to be there at that time. And I was the junior person on the Flagship team thinking about CRISPR technologies and what we could do. And we ended up starting this company, Editas Medicines, with folks from Third Rock Ventures and Polaris. Feng Zhang was involved, Keith Jung, actually Jennifer Doudna was involved in the early days as well. It was an incredible collaborative effort. And my task at the outset was to figure out what diseases we should go after with CRISPR. And I should pause to say this section, I'm not going to talk about some of the companies that I've worked on. The lesson here is to expect the unexpected. So spoiler alert. But the first task I had with Editas was to think about what should we work on with this technology. And I came in very confident and excited with all the tools at our disposal as a trained MIT engineer to try and figure this problem out. And attacked it in a very logical way. We thought about, well, the disease has to be very severe for someone to try a new therapy. It should be a monogenic disease because we need to know that the gene that we correct is going to be the driver of the disease. It should be expressed in a tissue where we have tools to be able to get the CRISPR molecules into those cells. And the result of the analysis essentially turned into some version of Editas' pipeline. I'm showing you the 2022 pipeline. So you have a set of diseases for ocular gene therapies, right? Indications in ocular gene therapy. Ex vivo, so you can do the correction outside the body, solve some of the delivery problems. Cell therapy and oncology. And in particular, the Edit 101. So this one, I love this one. It was Leber's congenital amaurosis caused by a single point mutation in the gene CEP290. And interestingly, this point mutation was in an intron, so a non-coding region of the gene. And what this point mutation did was that it basically introduced some sort of cryptic splice site. So you'd have this single point mutation that would cause the transcript to be truncated, and then there was an early stop codon, and so the protein was non-functional. Protein expressed in the retina, and so it would lead to blindness. It was a genetic form of blindness. But from a technical perspective, remember first generation CRISPR, we're thinking, wait a second, the correction here is not even in a coding region. Like you can sort of cut and you know, it's in the intron. All you need to do is just get rid of that mutation to destroy that splice site. About as perfect of an indication, I think at the time I could have thought of. We as a team could have thought of and come up with. So here's the pipeline 2024. My eye stuff is gone, right? What went wrong? cured people of blindness is what went wrong. This is the results of the phase 2 study of EDIT 101 in patients with LCA10 driven by mutations in CEP290. And the most correction they got, the 3 patients shown in the orange boxes, 2 of them were homozygous for the mutation, meaning they had 2 copies of the mutation. And for patients who were heterozygous with 1 copy of the mutation, the therapy was less effective. And so it was harder to predict. And so if you're trying to pre-select the patients that are going to be most effective, the homozygotes, the drug works better in them. Makes no sense to me, because you would think that if all you have to do is correct 1 copy, that should be easier. It should be harder to have to correct 2 copies. And yet, when the experiment was run, it turns out that you have to correct, if you can correct both copies, it works better in those patients. And there are not enough people in the world who are homozygous mutant in CEP290 to support a clinical program. Right? You can win, and doesn't mean you're going to win. Unexpected outcomes. This was supposed to be animated. Well, whatever. We'll talk about it. Rubius Therapeutics, another wonderful company that we helped to start. So this was work that Harvey Lodish had started at the Whitehead Institute, looking at culturing of red blood cells, and then with Hidapluo engineering the red blood cells. And we had Flagship came in and ended up building an incredible technology platform to manufacture red blood cells with therapeutic payloads, put those into patients. I remember when we started the project, there was even a paper that had come out recently, a review article from a well-respected academic that basically said, this is impossible. The technical challenge of manufacturing red blood cells is one that will essentially never be solved. And Rubius figured out a way to solve it, to the point of actually making large quantities of cells in industrial bioreactors, going back to some of the basics of sparging and impellers and all that sort of stuff, and really solved a number of technical challenges. And then the Fed increased interest rates. So you may remember that for a time, interest rates were at 0%. And suddenly, over the course of about 6 months, interest rates went from 0 to 5%. You may say, well, gee, macroeconomics, that's not what I trained in. That's not sort of a... Well, it turns out that macroeconomics affects all of us, right? So if your checking account or your CD ladder is returning 5%, the same is true for folks with institutional investments. And say, I'm getting my return in the safe assets, maybe I'll pull back on some of the risky assets. What could be riskier than preclinical biotechnology, right? So you start to see how that it affects publicly traded companies. It affected companies trying to go public. It affected private companies. It affected investors. The entire industry had this sort of shock effect from the increase in interest rates that led to, and continues to happen, but I think it perhaps is settling, but a number of companies that ended up having to shut down or close operations or otherwise divert from their paths. And so that's unfortunately what happened to Rubius along its journey. Here's another one, maybe a bit of a happier story. Gavor Grigorian came down to Flagship. He was a professor at Dartmouth at the time. He said, I've got this idea that there's a common language of proteins, that there's conserved features in proteins that can be learned and extracted from the data of all sort of protein structural biology. And at first I thought, well, gee, finally I'm back home. I did my research in protein engineering and finally a project related to proteins. But with Dane, we'd often talk about proteins are dynamic, they're wiggly. If we get a structure, let's not overinterpret the structure because there's protein dynamism that might be being masked by a single snapshot in time. And yet, so I was talking to Gevorg, I thought, well, if this is true, if there are actually structural features that you can extract and learn from and rebuild, you could start to generate new proteins that have never been seen before, and this would be really big. If true, it would be huge. And so we started working on it together, and Flagship and many other folks built this company, Generate, that seems to be my core belief, my core dogma, I did totally rethink for this. And one of the things that I'm most amazed about, I try every year to think about what's the thing I changed my mind about in the last year. And for this year, I used to be a skeptic of the power of AI applied to scientific questions. I think I'm losing my skepticism across the board in proteins, protein prediction, foundation models, genetics, small molecule world. There's just such incredible work being done at that frontier. I think that's going to be, that's a place I've changed my mind, and that as I think about my path from flagship, an unexpected outcome for me. Right? So we'll call that nonlinearity. Expect the unexpected, whether it's that you think you're going to win and you don't, macro conditions come in, any number of other factors, surprises, changing of your own dogma. It's worth to keep an eye on the unexpected. Check on time here. Perfect. Okay, let me tell you about Gate. This is the company that I'm currently the CEO of. So Gate is developing a class of small molecule drugs that we call molecular gates that essentially are small molecules to eliminate disease-causing extracellular proteins. And they do so at the source by stopping the cell from producing the protein. So let's see if I can get we got a laser pointer, great. So there's about 4,000 or so secreted and membrane proteins that exist outside the cell. And all of them get made inside the cell and have to transit through the secretory pathway. And it turns out that they all use a common channel, a protein called Sec61, or the secretory translocon, as their vehicle to get from inside the cell to outside the cell. And that's the purple diagram shown here. And so molecular gates are small molecules, so chemical drugs that bind to the channel and in a sequence-specific way stop a particular protein from going through. That protein gets redirected to be degraded inside the cell, and then whatever pool of that protein may have existed outside the cell disappears. So we think about diseases caused by extracellular proteins. Maybe a protein's produced at too high a level, or at the wrong time, or at the wrong place, or it misfolds, and you want to get rid of that protein. A molecular gate may be a way to do that. So that's the core technology of the company. The lesson here, I'm going to call it a lesson in self-assembly because I was remembering one of the qualifying exam questions I took, and it was had to do with the aggregation of hydrophobic species in a hydrophilic solution and how it's driven by entropy of water and the entropic cost, and that's driving the proteins together. And how despite the unexpected outcomes I talked about before, that's pretty immutable. Like you really can't solve that. And the reason I thought about that as the lesson here is that one of the diseases that we're going after with molecular gates is a disease that's very much a disease of protein aggregation, right? So it's a disease called prion disease, and you might be familiar with it from the mad cow epidemic of the late 90s. But the way the disease manifests is there's a protein that we all express in our brains and actually throughout our bodies called prion protein. And if it misfolds, it can misfold and expose a hydrophobic patch that when that bumps into another prion protein will cause that one to misfold and expose its hydrophobic patch and essentially trigger a chain reaction of misfolding leading to protein aggregation, neurotoxicity, and death. So it's a horrible disease. Typically patients, when they're diagnosed, have about a 9 to 12 month universally fatal rapid neurodegeneration, right? There was the epidemic in the 90s of eating contaminated meat, but that is tapered off with better food security. Nowadays, there's a genetic form where people are carriers of a mutation that causes their prion protein to be prone to misfold. And there's a sporadic form where you can just happen to anybody. And there's 12,000 people a year who acquire and succumb to prion disease. So our hypothesis here is that if we get rid of prion protein, that we would be able to stop prion disease. The genetics are such that people and animals who have loss of prion protein don't seem to have any adverse effect. And if you remove the normal protein, even if there is an aggregate present, there will have nothing to propagate the chain reaction. It's not our only hypothesis. There are a number of other companies with other modalities targeting the DNA, targeting the RNA, as ways to eliminate prion protein. And we're going after it at the level of the molecular gate mechanism, stopping the secretion of the protein. One of the cool things about working in drug development and drug discovery is that you sometimes get to meet the patients. And in this case, this project has been incredibly accelerated by folks who live just across the street here at the Broad Institute, Sonia Valab and Eric Minikel. And so it turns out that she's a carrier of hereditary prion disease. They've talked openly about their story. Her mother passed away, and they discovered afterwards that she died from prion disease, and upon genetic testing, figured out that Sonia was a carrier. And so Sonia and Eric, who at the time were not scientists, dropped their careers, came back here to Boston, trained as scientists, and opened up a lab at the Broad Institute with the mission of finding a cure for prion disease in Sonia's lifetime. Eric is an incredible science writer. He's at a blog, Cure FFI, that he's been writing for years. I used to read. And so I wrote him a cold email. It was actually before we had funding for GATE. I said, I've been following your story for years. I feel like I know you. You don't know me. But we have this idea about small molecules to eliminate prion protein. And what do you think? And so we got in touch and we began working together. We sent them some early molecules. They ran a Western blot and said, well, prion protein disappears. This is, they were very excited. And so we began working with the company to try and make molecules that would be selective and potent enough to be medicines. And over the now 2.5 years that we've been working together, that project has continued to the point where now, sharing some recent data here, we have molecules that we can put into mice that are potent and selective against prion protein. And as you can see, when we measure the amount of prion protein in the brain, we're knocking prion protein down 80%. You may say, why 80%? If we look in the colon, it turns out the prion protein goes down nearly completely, which tells us that probably it's an issue of those molecules getting across the brain. And so there's more work to be done on improving the pharmaceutical properties and really turning these things into drugs. It does teach us, interestingly, that maybe there isn't a floor to the mechanism. So always thinking about what we can learn from this type of data. But this has been really inspiring for the team, especially that we know someone who, if this works, it may really be able to help. But I'll tell you what's also been going on while we've been doing this. And I shelter the team from this, but as CEO, this is the type of stuff that I have to deal with. So interest rates go up, and people decide they don't like rare diseases anymore. And the IRA gets passed, and it has Medicare price controls that differentiate between small molecules and biologics. So price negotiation goes into effect 9 years for small molecules and 13 years for biologics. And whether or not that's going to make any difference, nobody knows, but sure as hell starts the room and the conversation about, well, gee, maybe we don't want small molecules anymore. And so you start to, as we build the company, there are all these external factors pushing against us in ways that it reminds me of the difference between a compass and a weather vane. Compass is telling you where you should go, and the weather vane just tells you what you might have to do to get there. These are really important lessons. Some things are immutable, and you should know what those are, but other ones are not. And so you should be a compass and not a weather vane. All right, last story. This job is very experiential. Whether it be building companies, working in biotech, anything you do really, it's highly experiential. It's instinct that you develop over time. You can develop that instinct by mass action and making these mistakes yourself. You could also maybe catalyze that reaction, have it go a lot faster. Right? And in the case of this type of stuff, I think the greatest catalyst is other people who can teach you things. So in my case, Dane, of course, we started and scientific intuition and ways of thinking about the world, but then also over the course of my move into Flagship, Nubar, and Avak, and others, and then throughout the companies, people who have been those catalysts for me in sharing their lessons. I was telling the students earlier, one of my mottos for my kids is make new mistakes. So this is a way to make new mistakes. You can learn from other people's mistakes, and you don't have to make them yourself. You'll also notice that some of the folks in here are relatively young. It's not all just of older, wiser folks. And my wife and my sister are both surgeons. And in surgery, there's a dictum that you see one, you do one, you teach one. And that part of the instruction of a craft, it involves all three. And in fact, the teaching one is a big piece of that. And so you'll be surprised how soon in your careers, as you start to leave MIT, you can become the catalyst for the people coming right behind you. And so I encourage you to be that catalyst as well for your community. So our time together is coming to a close. But I wanted to thank you all for the invitation, of course. It's been a great pleasure to be back. And I hope that some of the lessons I've shared and my stories can help be a catalyst for some of you on your journeys. It's been wonderful to meet the students and some of the new faculty members and hear about all the great work that's going on here. And I can't wait to see what you all create. So thank you. also be available afterwards
I
for.
was gonna say, I'm gonna grab the microphone and walk it around, because people don't want to walk, they want to talk.
Thank you, Jordi. Fascinating to hear the series of stories. But one thing you didn't tell us is what you might have done differently in retrospect. Because often we look back and say, I could have, I could have done that differently.
Yeah.
What advice do you have along this journey?
Yeah, there will always be things you wish you would have done differently and that you would try to do differently the next time. I think one of the, in that talking to people idea, it's not just saying hello, it's also asking the pointed questions and trying to figure out, for example, a concept I think about is frontiers of failure. We have ideas of how things might start, and folks who have seen them through and seen how they fail will understand what that frontier is, even though I haven't experienced it yet. And so being able to extract that information, seeking that information out, being open to the contrary opinion. If someone tells you something that doesn't fit with how you see the world, it's not an opportunity for convincing them. It's a gift, because they're telling you what they perceive. And you're able to factor that in. What an incredible gift to get that from people, and to factor that into your worldview. We'll always make mistakes, but yeah, as best as possible to try and minimize those and learn as many new mistakes as you can. This is the best.
Well, Jordi, that was a brilliant talk in my view. It was really evidence of what you said your special power is, which is to craft narratives. And a lot of people have gone through the journey you've gone through, especially a flagship, but very few would have taken these things away. So I think it's really useful for the students, and some of this will only become animated when they actually face the situation. So I remember lots of talks 40 years ago, not 18 years ago, sitting in this room and with older furniture and thinking, how is this useful to me? And then kind of being tempted to tune it out. But I'd say what they should do is really try to keep these things in mind and then apply it when the situation happens. But so a question I have for you, since you talked about leaping and thinking ahead, is you talked about your kind of changing views towards AI and what it might do in science. Just yesterday or today, OpenAI has been kind of talking about their most recent versions of their LLMs being basically comparable to a PhD in any given field, which is kind of a crazy notion, but that is what they say based on the reasoning tests they've done. Assume for a second that however you measure that is achieved. So two questions. One, what would a place like MIT need to provide as education or life tools for a world in which that's the case? So if you think ahead. And then, of course, to the students in this classroom. And then in such a world, if you imagine it would exist, how do your convening, leading, kind of the social power of human interaction to create new things, what role does that play when you have machines on your team? You knew it wasn't going to be easy.
I had 8 years, Nubar, of being in meetings with you, and everything would go really well, and then you'd say, oh, one last question. And I'm feeling all those emotions coming back in this moment. So thank you for making me feel young again. So there's some sets of tools I've come across recently that are sort of AI, LLM type of tools around scientific literature. Right, we ask a question, is there an association between this target and this disease? Or what happens if I eliminate protein X? It spits back an answer with citations. And I look at that and say, that's my job that is just taken. Because it was that ability to, having a breadth of therapeutic understanding or mechanistic understanding, and be able to apply that understanding to know what questions to ask of PubMed, that some of these tools are automating. Puts me a bit on the side of the dinosaur here, but I think for the next generation, to have that tool at your disposal enables you to then ask and then query the next. And I think what it might push us toward is actually building of solutions that are perhaps better tailored to the problems that those solutions can solve. So one of the challenges relatively universally is, I built this tool, what is it good for? Well, it's the thing, I should do it for finding the product market fit in the tech world or finding that solution. I wonder if perhaps the ability to query solution space and figure out that answer through AI may in fact then direct the builders to know what things to build for that, perhaps. Second question.
How does your social
Ooh.
approach work?
The social disconnect that has happened since the advent of social media and the internet and the balkanization, siloing, and isolation that has ended up in the pandemic of loneliness or the epidemic of loneliness everyone's talked about, I think the answer is going to be in hyper-local communities. I think there's something about where we live and the basics of life that require other people around us. And so I do think that core human connection will always be there. What these tools may allow is for those connections to take place over much greater scales than they currently do. So I can tell you a story about the starting of Gate. It was in the depths of the pandemic. I had all 3 boys at home. We were doing kindergarten over Zoom. It was nuts. And the 3 of us co-founders, who I showed you the picture of, were in 3 different places. We had California, Colorado, and Oregon. And yet together we started working on the company. We never met in person until months and months later, when we were very close to having the funding in place. Investors being willing to meet and trust and invest without having the face-to-face meeting with a handshake. That wouldn't have happened but for the pandemic and the social changes, as we were talking about earlier, Chris, that went along with that. So I've got to believe that we as humans will adapt, that there will always be a need for that connection, and thus the ability to tell stories and motivate people and lead. It just may happen at greater scales and different times than we currently see it.
Okay, so hi, I really enjoyed your talk. So a couple of things that struck out to me that compelled me to ask a question is, you had said that so what is a lot of times more important than the what when it comes to being a business. And I think that's why I think that biotech is so interesting, because I think at the end of the day, it's meant to respond to needs and to especially in the medicine space, care for patients.
Yep.
But something that you had said was, particularly with prion disease, how I think it was 1,200 people are diagnosed, or a year, it might have been 12,000, okay, my bad. 12,000 people are diagnosed a year, and obviously there's no cure for it at this point. And it makes me think about how do you reconcile being an engineer who cares about patients with being a business person, because something like this, it's hard to quantify value, because it's it's a very small pool of people who are affected by it, but obviously it's so serious because there's no cure. So it's how do you hold fast to that motto that the so what is so important, but it's if the economics are not in your favor?
Yeah, you know what? It's a great question. It's easier than Nubar's question, by just a tad, or at least I have some thoughts on it. Thank you for asking it. With the prion case in particular, I think of it like poetry, actually. Right? You can write poems in any way you want, or you can write a sonnet. And a sonnet has a set of rules around it. A certain number of quatrains and a couplet and a certain number of syllables. Why? I don't know. It's restriction. It's restriction on the form of the poem. But you see it throughout the arts. Artists will put restrictions on themselves. I'm only going to use a certain type of antique equipment, or I'm only going to write poems that fit this model. And then you can be creative and create art within those restrictions. And so the way I've dealt with it personally is to think of some of the headwinds, not just in the prion case, but throughout, as the rules of writing the sonnet. It's okay. It's not a matter of bang your head against the wall. Oh, I just need to make a better argument. If only I showed them this data. No. Okay, this is what the world is telling me. These are the rules I have to follow. Let me go follow, find a way to make the solution that fits those rules.
Hello, thank you. It was a really amazing talk to see as an undergrad. What was your experience stepping from academia into industry? I think as an undergrad, it's very easy to fall into the trap of the sunken cost fallacy of like, I'm undergrad already, I'm at MIT, I might as well go to grad school, I might as well try and be a postdoc. What was that step forward like for you, and what were some difficulties you found?
I think having the imagination to know when to step off certain paths or follow certain, maybe imagination, intuition, to know when to follow those paths is something that you'll develop for yourself as you go. I think going into graduate school can be incredibly enabling and powerful. Everyone in this room, of course, but also sort of what it teaches you about how to do inquiry, right? Preparing you for experiments that are not going to work. And so you have to have thought ahead about what you will do when that experiment doesn't work, right? And the creation of new knowledge and not just the acquisition of existing knowledge. So I think that's all very important stuff in grad school. Whether you go on the academic path towards being a professor, or whether you take some alternate path into whether it be biotech or any number of other patent law or whatever it might be of alternates, is about you figuring out what you actually want and are motivated by, or that fugacity concept. Because you might find that you love doing the research and you love teaching. And if that's the case, great, then that is the right role for you. But the more you can define that for yourself, and the more open-minded you are about not saying, well, I've started grad school, therefore the only successful outcome is right? The more open you are there, I think that maybe gives you the best shot to find your best landing spot, or to create it for yourself, because it may not exist yet.
Hi, I really enjoyed your talk very much as well. Are you worried about the bias within AI that no one really sees or understands or can identify until it's too late?
Certainly in social uses of AI,
in
absolutely.
everything.
In
everything.
Absolutely. I think scientifically, the risk, it's a different risk profile. So for example, if it's a medical algorithm that is improperly recommending people for therapy, or improperly flagging people for therapy because of biases built in, that's a big problem that needs to be solved. If the algorithm is spitting out a protein structure that looks like this when it should look like that.
Well, you just mentioned that. If I can just give you a follow-on question. It's been long-standing accepted that the key to being able to predict structure is by looking at the sequence structure relationship, right, and function. When I think it's been very much overlooked to think about weak interactions in proteins. And that's really what is defining stability and structure, as well as function, because it defines the interactions between proteins and other drugs. So this is a new way of thinking about this, but the tools aren't really available, and so you're generating them, and you're trying to prove this to the community. AI is going to be very fast, very describing all of these things because it's the accepted knowledge. It's out there and it's published. So how can that come to one?
Yeah, if I can rephrase the question back at you, is it perhaps these models trained on what they've seen before, will they be able to predict things that they haven't seen before?
There are underlying governing elements that haven't really been defined yet.
Will they be able to tease it out? So I don't know the answer. I think the optimist's view is that the model will be able to extract things, even if there's no underlying understanding of it, that in the best possible world, we as humans will be able to see and then interpret. I'm intrigued by some of the work that's being done to take neural networks and large language models and find a way to have them output what they are doing in the language of mathematics. Whether there's any fundamental truth or sort of actual reasoning going on, today I'm still skeptical. I'm not an expert in the field. But I think the ability to take whatever that is and spit it out in equations that we can then use as intuition might be a way forward to really unlock that.
So I have to say, based on Jordi's background, I did not expect fugacity to make an appearance in the presentation today. So this is how we know you are still a chemical engineer at heart. Once a ChemE, always a ChemE. For all of you who are thinking you may do something as exciting and maybe a little bit unexpected as Jordi has done. Thank you for spending the time with us. Thank you for sharing your lessons with us. I am sure that, as Nubar said, many of the people who are listening to you today may not know that they heard what you said until later when they're applying it to their own lives. So thank you very much for sharing your experiences with us.
Great. Thank you. This is great. Yeah, that'd be wonderful. Oh,
Okay.
sure.